afternoon, everyone. Thank you so much for joining us for the Transcend Research Seminar. We are very excited to have Dr. Katie Hinde come speak with us. Dr. Hinde is in the School of Human Evolution and Social Change. Before she was at Arizona State, she was at Harvard, I believe. I'll let her do a full bio, but we're really excited. She spoke for us last year, and she was so popular, we asked her to come back again. So we're really great to have her back. Thank you so much, Meg. Meg's being really nice about this. I got only a third of the way through my talk last year, and so I've been invited back to speak about like maybe the next third. Who knows how far we'll get into it. But basically, at any point, if you have any questions or you want a clarification or anything else like that, let's make this a discussion and not death by PowerPoint, okay? I want to start, see if this does it. Nope. 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 One second. Oh, yes. All right. Okay, well, counterintuitive. All right. So, I oftentimes present my work at clinical conferences, and the practice generally is to have a disclosure side in case I'm paid by industry or some other source of funding, that you should think about how that might influence how I am motivated to do my research, how I design my research, what I choose to research, and things like that. I actually am not paid by industry at all, and all of the grant research support for the work I'll be showing you today has come from the National Institutes of Health, the National Science Foundation, funding from Arizona State University, and when I was before at Harvard University in their Department of Human Evolutionary Biology. I do want to disclose, though, that I am an evolutionary anthropologist. And how many people here have taken an anthropology class? Awesome. All right. I strongly encourage folks to take classes in anthropology. Obviously, I'm biased, but anthropology is a field. Hey, Dan. Anthropology is a really exciting field because it sits right at the intersection between the life sciences and the social sciences. And for those of you that are nutritionists, like you also sit in that same kind of space, right? So you have to have a lot of knowledge about biochemistry and digestion, physiology, some of these, really, really bench sciencey kinds of things. But you also have to think about what shapes the food people eat, how they decide what to eat, or what they are constrained into eating, aspects of food deserts, or rather food apartheid, cultural practices, cultural influences. And so spaces where people are thinking both with life sciences and social sciences hats become really, really important, because that's really fundamentally how they improve human health, right? So life scientists can design vaccines, social scientists get people to actually use them, okay? So from an evolutionary anthropologist background, that means that I'm really fundamentally interested in how humans are shaped is a function of both our shared features with other primates and the ways in which we've had our own derived evolutionary trajectory within the human lineage. And so here you have, of course, illustration of the living great apes and lesser apes. Okay, so you've got humans, chimpanzees, gorillas, orangutans, the lesser apes, the sea monkeys and gibbons over there on the far side. But this is what is alive today. And I think it's really important for us to think about that humans are one of a multitude of hominin species that have been roaming different landscapes. So we're the last hominin standing, likely because we have been very, very good at turning food into babies and outcompeting other hominin species, but these are our not living relatives. And I fundamentally think about mother's milk and infant development at the lactation interface from an evolutionary perspective, right? That natural selection has shaped the adaptation of milk synthesis, that it has shaped adaptations within the mother to produce the milk, that it has shaped adaptations within the infant to use the milk, And this is where things get really nuanced, because if you are following public health messaging, you will hear that for most babies, compared to any other options, breast milk is the most optimal food for a newborn. That is accurate. But something being the most optimal among options is not the same thing as something being optimized. So, and the reason that it is not what we would consider to be optimized is that if we think about optimization, we think about what is best to support maximal developmental trajectories for the baby. Now, if the mom were producing milk at that level, she would quickly deplete her own body stores of minerals, of vitamins, of fats, and she would risk her own health. And so what you find then is that milk is a negotiation in between a maternal optima and an infant optima, and it can vary between them. It is exceedingly difficult to study. So, because that oscillation can actually vary across time or within particular time units. All right, so I just said a lot of stuff, but optimize is to make perfect, effective, or as functional as possible. And we know that mothers are going to be constrained along a number of parameters. And even if you remove those constraints in a single time point, natural selection has shaped mammalian mothers not to optimize the outcome of any one particular infant, but to shape a successful lifelong reproductive career. So natural selection favors the production of the most surviving and reproducing offspring, not the outcome of any one particular infant. This becomes a really difficult thing in clinical settings where the entire clinical team and the parents from a cultural perspective would like to optimize the outcome for that one particular infant, but it can present challenges from just a physiological constraints perspective. Maybe, there we go. How many people here have heard of lactation failure? Hands high. All right, so about 5% of the people in the room, which is about the rate of lactation failure in a clinical setting in a high-income country. Now, 5% of moms are going to have significant challenges to breastfeeding. They're going to have either difficulties with latch, difficulties with milk production, milk letdown, problems with the baby, being able to suck and have that neuromotor control to be able to effectively nurse, in which if that demand's not there, that's going to affect the milk production. So there's lots of pieces to lactation failure. It happens infrequently, and that's something that is talked about in clinical settings. But what isn't talked about is that the capacity of the mammary gland to synthesize milk actually has oftentimes a normal distribution, right? So here's a normal distribution, and you will have some moms that are going to be underproducing the milk that they synthesize. And no matter what you feed them, no matter how much you chillax them, and no matter how much you support breastfeeding practices, the moms are still going to have a ceiling on the amount of milk that they can produce. And some of these things are actually highly predictable, right? So we know that very, very young teenage moms are going to have limited milk production. We know moms that are really underweight are going to have underproduction. Even if you increase their nutritional intake, they're going to put body stores on their body at the same time that they even increase the milk, but you're not going to get all of that energy intake to be shoved into the milk production. Okay? You're going to have moms that are very, very overweight are going to have trouble because of hormonal dysregulation. And it is in this area of underproduction that we actually know very, very little from a nutritional or clinical space. Most all of the work is done on either lactation failure, getting milk production up and running for moms that have preterm births and have babies in the neonatal intensive care unit, or just everybody else kind of gets lumped together. There's deficits in this research area. I touched on this a little bit earlier, but it's really important, I think, whenever we're talking about organisms and biological systems, that we recognize that fundamentally at cellular and larger levels, there's always going to be trade-offs, right? So like if you spend $5 on coffee in the morning, that $5 is not available for croissant in the afternoon, right? You spent it, it's gone. Same thing's true with calories or skeletal calcium, right? So once you allocate those efforts to one space, it's not available for another investment, okay? So we see routinely variation between mother's condition and lactation, right? So moms that either currently or in the recent past or during development had caloric restriction, that's going to affect their ability to synthesize milk. We're going to see aspects of the mother's, I mentioned teenage moms. Teenage moms themselves are still developing. So 16% of all babies born every year around the world are born to teenage moms. And they're still growing. So energy that goes toward lactation is not available for their own growth, which can stunt their growth attainment and impact reproduction for the rest of their lifespan. So there's going to be trade-offs there. You have the quantity versus condition of babies. So when you have twins or triplets, the total fetal placental unit is much bigger, but the individual babies are on average smaller, right? Everybody good with this? It's super, do you guys all know all this? Should I go faster? Maybe? Just keep looking at the monkey pictures. And all of this is shaped on a temporal scale, okay? So natural selection favors attributes that shape a reproductive career, which is made up of each individual reproductive event. So overinvestment in current reproduction can compromise conditions, survival, or resources available for future reproduction, right? So there's going to be trade-offs across time. And I always show this slide. In these systems, you allocate resources like energy, time, or calcium to staying alive. So cellular processes for maintenance, thermoregulation, tissue repair. Anything that you have over that early in life, it goes to growth and development. And if there's constraints within the system, organisms will prioritize staying alive over growth and development, which is why you'll see that children, when they get sick, their growth falters. And you can see this on weekly level. As mammals, we hit a point where we prioritize adult activities above growth and development. We're not trees, we're not sharks that can continue growing throughout adulthood. We transition from a period of growth and development to prioritizing reproduction. Now, this trade-off matrix early in life is shaped by the mother's milk. And so I already said that, I say that all the time, and not necessarily optimal for any one infant. And when an individual is developing among mammals, that constraint within the system in a natural ecology is going to be the mother's breast milk. So if the mom, however much calories, calcium, vitamins, minerals, as much as is going in the milk, that is what is shaping the infant's developmental priorities. And that baby is going to have to grow. And in nutrition and public health, we spend a lot of time thinking about somatic development, how a body develops. But we are not the last hominin standing because we were the biggest hominin. We're the last hominin standing because we are really, really smart, really, really cooperative, really good at solving problems, really good at doing all sorts of behaviorally flexible and cognitively challenging tasks. So when we think about early life development, there's going to be prioritization for some extent on attaining a certain threshold size for survival, but there's also going to be a lot of prioritization of development of the attributes that fundamentally make humans human. Okay. And so this is a research area that I'm really focused on, and that's understanding the way that mother's milk shapes things like temperament, behavioral activity, and now neurobiology. So I'll be spending most of the rest of my time talking about those domains. Any questions about trade-off or life history theory? All right. Okay. And I want to highlight that work in this area on how milk is predicting these aspects of infant development have been conducted in humans, rodents, monkeys, and piglets. Not all by me, but in this domain of research. And so if you have any questions about some of the species I don't cover, I will do my best to answer them. All right, oh, sorry. This thing is really hair trickery, so I'm gonna try and go a little bit slower. Okay, how many people here have heard of glucocorticoids and cortisol? High and proud, come on, yeah. All right, how many people think of cortisol as the stress hormone? All right, at the end of the slide, I'd really like you to take a picture of it, because if it's the only thing you learn from me, I will give up life history trade-offs for you to learn about like cortisol and its magical complexity, okay? All right, so cortisol is a heavy lifting hormone that does all sorts of instrumental things for our physiology all the time, every day, okay? So it has key functions for the endocrine system. It can be pro-inflammatory or anti-inflammatory, depending. It is really important for neural tissue health. So people here, you guys know the Goldilocks fairy tale, the like too hot, too cold, just right. You know, the little white girl that broke into the house and like took all the shit and like, you know, great story for our kids. Anyway, just the right amount of cortisol can be incredibly important for forming memories, for forming connections, for inoculating neural tissue to function very, very well. If it gets swamped in cortisol, it can become neurotoxic. And if there is not enough stimulation of that system, it can leave the system unable to effectively respond to challenges later on, okay? It has a lot to do with bone turnover, bone growth, connective tissues, and muscle integrity. It is really important for our metabolism, right? So glucocorticoids are important for telling the body to store energy, how to store it, where to store it, and then when you need to mobilize it to use it, how to pull it out of storage. And there's as yet not well understood but important interactions with the microbiome of the intestinal tract. So when the host gets really, really challenged and stressed, that's going to change the microbial community. The microbial community can release neurotransmitters like serotonin and dopamine that can improve mood and buffer the host from certain stresses. So there's this really complex biofeedback there, of which the HPA axis is implicated. Everybody knows what the HPA axis is, right? Oh, okay. Thank you. Thank you for signaling that you don't know something. All of your colleagues that were too nervous to do that, they appreciate you very much right now. The HPA axis is the hypothalamic pituitary adrenal axis. Okay, so two tissues within the brain and then your adrenals that sit right above your kidneys. And this is a hormonal cascade that communicates downstream, stimulating the adrenals to secrete cortisol to then do all of these things that we've been talking about. That cortisol then travels through the blood-brain barrier, binds to receptors within the hypothalamus, and turns the system down, right? So it's a negative feedback loop. So you have stimulation, you secrete ACTH, you secrete CRH. CRH travels to the adrenals are like, got the message, let's secrete some cortisol, and then it goes back to the top and turns the system down. That's a negative feedback hormonal system. Can anybody think of a positive feedback hormonal system? I hate when I ask that question that way because you might think of one and I don't actually know it. So I'm just going to give you the answer that I was hoping somebody would say, which is a positive hormonal feedback system is the oxytocinergic system, right? So there's secretion of oxytocin. The secretion of oxytocin stimulates more secretion of oxytocin. Do you guys know what oxytocin does? It is love. Oxytocin is a deep ancestral mammalian hormone that underlies the biobehavioral bond between a mother and her infant. So it is released when moms let down their milk, which is stimulated by prolactin, but then there's an interaction that releases oxytocin too, and that causes contractions within the muscles of the mammary gland to help push out the milk for when the baby is nursing. So oxytocin underlies the first social relationship within the mammalian lineage and within a mammal's life between the mother and the baby. So all of the other things that oxytocin does, trust, friendship, orgasm, social bonding, romantic love, all of those things came downstream from the original adaptive trait of mother-infant bonding. So we could not be cooperative and complex socially to the extent that we are in many ways if it weren't for that original social bond. Okay, all right. Oh, also cortisol does stuff for carbohydrate metabolism. Okay, so always gets grumpy when I walk away. All right, now I'm just going to tell you a couple things because they're not in this particular slide deck. We think of cortisol as a stress hormone because it spikes really high when we have a really challenging experience, okay? You get into a fight with a loved one, have a car accident, have to do an exam, have to publicly speak if you don't like public speaking. All of these are jump out of airplanes and go skydiving. All of these are going to stimulate your HPA axis for this really high bolus of cortisol. But let's just say you're having a regular day. It's your routine day. When is your cortisol likely to be the highest? In the morning, yes. When in the morning? You know, that super stressful time, 30 minutes before you wake up and you even know consciously that anything's going on, right? No, right? You're asleep. Your cortisol spikes before you wake up to wake you up. It helps accelerate your heart rate. It helps raise your body temperature. It helps you wake up to embrace your day. That is part of what cortisol is doing. It fluctuates according to a circadian rhythm across the 24-hour period. Okay? So in a normal day, your cortisol is going to be the highest before you wake up. Now, do you guys know who doesn't have a circadian rhythm? Babies. Babies don't have a circadian rhythm. It starts to develop partway through their first year postnatally. And right now, we don't have any empirical evidence of how that circadian rhythm in babies gets trained. So is it something that just automatically gets turned on genetically at a particular developmental stage? Is it something that's guided by the bioactive hormones in mother's milk, the cortisols in milk? Moms that breastfeed throughout the day and night, the baby is going to be getting a cortisol snapshot of the mother's physiological regulation. So when her cortisol is high, the cortisol is high in her milk. When her cortisol is low, it's low in her milk. So a lot of us have speculated that that circadian rhythm of hormones in breast milk is important for onboarding the infant circadian rhythm, but nobody studied it yet. Please fund many submitted grants. So I do my research. I'm really interested fundamentally in humans, but humans are very complicated. So what are some ways that humans are complicated for understanding these adaptive dynamics of lactation biology? Anyone? Okay, so humans have very complex nutrition. And then does everybody eat the same thing? Right, so you've got high dietary variance from individual to individual. I'm pretty sure you're talking about mom, right? Okay, but like, is there nutritional variance for babies and humans that's different than other species? Like, is there something that we can buy at the store that maybe isn't breast milk? Right, so we've got formula. So a lot of moms use formula, and there's a non-random distribution of who is more likely to breastfeed and who is more likely to formula feed that can complicate our interpretations of those data. So I do a lot of my research in rhesus macaques, which are the #1 biomedical non-human primate model. Anybody here ever see a rhesus macaque? Yeah, they don't tend to have them in zoos because they're super jerks. They are really aggressive. They killed the deputy mayor of New Delhi in 2006. 3 of them worked together to throw him off a balcony. They are very smart. They will cut you. And yeah, they will cut you. And all right, so like, did I do this last year? Did I tell people how to survive a monkey attack? Do you guys want to know this? You know? All right, first of all, do you know how to survive a dog attack? Sacrifice your arm. Okay? A dog's aggressive, lethal technique is to bite down and rip and mangle. Okay? So if you sacrifice your non-dominant arm, you can punch them a whole bunch of times in the nose. This will cause a huge amount of pain. It will make their eyes water, which they perceive as a huge vulnerability because they can't see as well, and they will release you and back down. It's your best chance. Okay? Don't run. That is super risky. Okay? If you try to do the same technique of sacrificing your arm to a monkey, what they would do, so monkeys, so cool, so cool. Every time an adult male monkey opens and closes its mouth, its upper canines literally sharpen on the whetstone of their lower canines. They are as sharp as blades, and they fight like they're swords. So what they do is they slice down to the bone and then launch past that arm to your face, okay? So if you start to get a monkey that's being really aggressive to you, again, don't run. That is a super strong signal that they can take you down to things that are really aggressive like that. What you have to do is you have to scream and punch back and protect your face, okay? Those are your best options. I had a monkey come after me in Indonesia, and I threw my clipboard and my binoculars and everything at its face and screamed at it, and it sat down and walked away. Everybody that tangles with the same monkey that like ran like got chased and sliced. So monkeys have sharpening mechanisms in their teeth. Dogs and felids and other kinds of predators don't have that. Monkey fact. All right. So it's important to recognize that there are a number of things that make monkeys not an ideal model, some of which I have said, but they are very expensive to work with in captivity. The experimental manipulations you can do with them, I think, depending on your ethical approach to animal science, are going to be variable, and they live a really long time. So even though the rhesus macaques I work with develop and mature 3 times faster than humans, so I'm now working on my 3rd generation of monkeys, they're really nothing like a rodent where you can have like 100 generations in the same amount of time that I've gotten my 3 generations for monkeys, right? So you have some trade-offs there. Some positive things though are that they have a lot of similarities with humans. So their milk composition and the volume of milk they make is very similar for their body size. They have slowly developing singleton infants. They are characterized by very extensive, very nurturing maternal care, and they have complex social behavior, and they have a lot of individual personality. So a lot of things that we think of as making humans very special, we share with monkeys. Not everything though. So rhesus macaques have, so this is, we collect milk at early and peak lactation, and there's some very specific behavioral development things that happen between these 2 time points. So at 1 month of infant age, that first month, the baby's pretty much on the mom all the time, always having access to nipple, not really locomoting or moving around, not really interacting with other individuals than the mother. After that 1 month time point, they start to play, they start to explore, they start moving away from the mom and returning to her as a secure base. For anybody that has followed like Bowlby Ainsworth's work. And we know that underlying these behavioral developments are very specific changes that are happening in their neural function and their neural architecture. So we know that in human babies and the monkeys, that after birth, there's this incredible period of neural proliferation. The number of neurons in the brain vastly expands. And then that proliferation is pruned back and canalized for more effective electrical signals around the brain. And this is typical neurodevelopment, and how, this is why they say that how animals or humans are interacting, how they're behaving, what they're experiencing, that shapes the proliferation and the pruning back. So this is a really important period of neurodevelopment. I've been doing a long-term collaboration with my colleague, John Capitanio here. He's the tall one. Sally Mendoza started the field of biopsychology at Stanford in the 70s, and Nancy Capitanio is also really fun to hang out with. So that's all of us out to dinner in Northern California. And, okay, back to the science. What we do with the baby monkeys is that 3 to 4 months of infant age, they come in for biobehavioral assessment. This has been done on actually now over 5,000 baby monkeys. We know the heritabilities. We know all about these babies. And at the end of this 24-hour baby assessment, they're all rated for their temperament along these trait adjectives. And we're really interested in their confident temperament and their nervous temperament. So confident is how confident, bold, active, curious, and playful the babies are. And the nervousness is how nervous, fearful, timid, not calm, not confident they are. Now, when you hear words like confident, bold, active, curious, and playful, what's something that comes to mind? Like anyone, like if you think of something as being like playful and rambunctious, No, I'm trying to, let me think how I want to ask this. Do we think that this is something that takes a lot of calories or not many calories? Right? The nutritional intake required to support a large amount of behavioral activity is higher than like mellowness, right? Okay. So we were quite interested in understanding how milk calories predicted behavioral outcomes. And what we found, sorry, I'm just gonna, how long am I supposed to talk for? All right, I'm not gonna show you that data, and I'll just tell you what we found. What we found is that moms that produced more milk calories at that 1 month time point had babies that were way more behaviorally active later on in infancy, independent of the milk that they were making at the time that we assessed their behavior later in infancy. And this was the first suggestion we had that mother's milk is organizing the behavioral repertoire, the behavioral phenotype. Okay? But we were also really interested in how hormones like cortisol might be implicating this, and I'll come back to why. So cortisol varies very highly in milk. These are the concentrations of cortisol for over 100 moms at early and peak lactation. And what we found is that when milk cortisol was higher, it predicted an infant that was more nervous and less confident at 3 to 4 months of age. And this finding was specific to daughters, okay? If milk cortisol was increasing from early to peak lactation, we found that this was associated with more nervous, less confident sons, okay? Wait, I can't remember what my next slide is. I'm gonna sneak forward to the next slide to see if it's useful. It is, okay. So what you're seeing here is this is a scatter plot. These are the residual values after we control for other important factors. And what you find is that as cortisol goes up, the nervous temperament rating goes up. And this pattern is true for both sons and daughters. But daughters were more sensitive to cortisol early in lactation, and sons were more sensitive to cortisol at a later time point. And these data suggest that daughters and sons have different windows of sensitivity, and that daughters' windows come earlier. And I feel really confident telling you about this because we now have seen supporting evidence for that in human data, and we've replicated it again in a different population of rhesus monkeys. The confidence goes down. You'd expect that. High nervousness and low confidence co-vary pretty strongly in the monkeys, right? So this is the work that we did with Amanda Detmer. Actually, she's at the Society for Neuroscience right now presenting the follow-up to this work. And we found that higher milk cortisol was associated with less impulsive and better cognitive and performance. Now, we only had 8 monkeys, so we weren't able to look at sons and daughters independently. So that's why this is purple. But we were also able to look at their social behavior. And what we found is that higher milk cortisol was associated with more social behavior for daughters, not for sons. Okay, and here are those data. So you can see that the black circles are daughters, and there's a positive correlation that as cortisol goes up, their total play goes up. And the sons stay pretty flat. They're not responding to the cortisol level. Now this is, in this study, we were only able to measure cortisol in the first month of life. We weren't able to look at it at later time points. So it could be that sons are sensitive to a time point that we weren't able to collect data on because she couldn't do peak collection like I did in my population. So this suggests consistently that daughters are more sensitive to milk composition in the first month of life. Now, we think that this has to do with sensitivity of sons and daughters because there actually isn't any difference in the amount of cortisol in milk for sons and daughters. So the absolute amount or whether or not it increases across lactation, it's the same for sons and daughters, all right? Okay, sidebar, I have to tell you something else that's going on that's really interesting about monkeys. They make different milk recipes for their sons and their daughters. We think humans do too. So here you have you guys seen violin plots before? Violin plots, but yeah. Okay, so they're different than a bar graph because the bar graph really just shows you the mean and then the variance around that mean in a standardized way. But violin graphs, basically they tell you where, they smooth out where all the data are falling, right? And they're called violin plots because they are shaped like weird musical instruments. And so what you see here is that the average amount of milk that's produced is lower for sons than it is for daughters. But the amount of variation for sons is much greater. So, can I step away from the microphone? I'm sorry, Corey. What you find is that the moms that are producing the most milk, they're raising sons. But on average, more moms with sons are making lower volumes of milk. But the consequence, some of these look more like vases or vases, however you pronounce that. Still called violin plots though. What you find, so this is the energy density of the milk. The energy density comes from how much fat, how much protein, and how much carbohydrate is present in the milk. And what we find is that moms that have daughters on their first infant are producing the least caloric milk. It's low in fat, it's low in protein. So the total density of calories for those daughters is lower. If they have daughters for their second born or at any point that they have sons, they make a richer milk. When we put these things all together, what you find is that the total available milk energy is still slightly higher for daughters. Okay? This is also true in dairy cows. And Megan Azad, who's coming in a month, November like 22nd, 21st. She's amazing. You guys should definitely come to her talk. She's incredible. She's also very nice. She's Canadian. I'm hoping that she'll present some of our data that we have on human cohort studies because she and I have 15,000 human subjects and yeah, human data sets. Really nice. We can't get their milk and we don't know differences in the milk production, but we would expect to see differences in breastfeeding behavior. So if moms are having a harder time synthesizing volumes of milk, they perceive that they're not making enough milk, we would expect moms of sons to introduce formula at younger ages. We'd expect them to introduce solid foods at younger ages, and we'd expect them to wean earlier. And we'd expect them to seek treatment for milk production like domperidol. And I listed all 4 of those things because we find all 4 of those things in the data set. Okay, so moms of sons and moms of daughters are equally intending to breastfeed at the time of delivery in the hospital. They say equal numbers, yes, I intend to breastfeed, or no, I'm going to formula feed. So even though the intentions are the same, we see that moms are more likely to formula feed sons. They're more likely to introduce formula at younger ages. They're more likely to introduce solid food at younger ages. They wean them several weeks earlier, and they're more likely to seek treatment for perceptions of inadequate milk supply. Okay. So our working conceptual model, I can do this, it's gonna happen. Our working conceptual model was that milk organizes in part infant behavior and temperament. And of course, we think that behavior and temperament are also going to be shaping how babies are nursing when they demand milk from their mom. And that together, these things are going to influence how individuals behave and how their brains function as adolescents. Okay, so this term neuroenergetics, it's a real term. It's a term for glucose use in the brain. So one of the primary methods of neuroimaging is a PET scan, right? How many people here are familiar with PET scans? Anyone? One of the most typical things that they measure is glucose uptake in different regions of the brain. What is glucose other than energetics? It's in the brain, so neuroenergetics, real word. So we predicted that with these sex differentiated windows of sensitivity and magnitude, we expected that the neuroenergetics of sons and daughters would be differentially sensitive to early and peak lactation. And we thought that temperament, as organized in infancy, would predict these things. So we really thought milk was going to be most impactful on temperament, and that temperament would impact neuroenergetics. Okay. What we thought. Okay. So we were interested in particular brain regions like the amygdala, the nucleus accumbens, anterior cingulate, caudate putamen, hippocampus, prefrontal cortex, and hypothalamus, because in monkeys and humans, these brain regions have all been implicated in fear and anxiety, decision-making, emotion regulation, social engagement, confidence, extroversion, memory, risk-taking, impulsivity, things that go along with nervousness and confidence. All those terms are up there is because everybody calls the same kinds of phenomena different terms, because we want to make things very difficult for people reading the literature. Okay, so in order to do this, what we do is we do a PET scan, and we look at glucose uptake within different brain regions, right? So the warmer, redder the color, the more glucose uptake is happening. We also do an MRI scan so that we can actually see the brain structures. We then go into the MRI, and using a brain atlas, we hand draw the outlines of each of these different brain regions on the MRI. This right here, sorry. Thank you for the and amazing husband. And the cognitive putamen is really fun because it looks like Pac-Man. And it's always the easiest region to draw. And then using software, you put the 2 scans together so that you can extract how much glucose uptake happened within the particular margins of the brain structures that you're interested in. Okay? So like, it's basically MS Paint. If there's 3 of us in the room that are familiar with that program, maybe. All right, so these are all the different views of a brain, so that you can kind of look at how the brain is oriented. And there's a lot of projections among these different areas that are communicating with each other, midbrain regions that underlie behavior, motivation, and emotion regulation. And for our study, we looked at 35 subjects because these kinds of studies are stupid expensive. I think I think I wrote that in my grant. We closely monitored the babies when they were born in 2012. We collected information about their mom and their behavior and the milk production. We did the neuroimaging right as they were ending adolescence, right before adulthood. And the technique that we used for the PET imaging was identical to the biobehavioral assessment that they had received in infancy. So they basically were put to the exact same challenge as adolescents that they experienced when they were 3 months old. And because the study was stupid expensive, we prioritized subjects that were characterized by the most divergent behavioral phenotypes. So the highest nervous and lowest nervous, and highest confident and lowest confident. Because where you expect there to be big differences, you expect that you'll have to be able to detect them with smaller sample sizes. And I'm going to tell you a story of science with these next slides, because my findings are going to be really, really exciting, and like, hold the phone, we're going to submit to a major journal. And then we thought harder about our data, and we're like, oh, we need to make sure that this isn't what's going on, and then that was what was going on, and then our findings are, we're going to submit to a typical journal. Okay? So this is why really you should think hard about your data, always. So what we found is that, and this slide didn't get updated, but got the newest data last night, so not all the slides are up to date. But in general, temperament was only weakly associated with the glucose use in these brains. So the temperament that they evinced at 3 months of age was not strongly predictive of what was going on later on. But when we first ran the data, what we found is that across all brain regions, for both males and females, so if there's 2 symbols here, it means it was in both hemispheres. If there's 1 symbol, it was just in 1 hemisphere. What we found over and over again was that there was higher glucose uptake in areas of the brain that underlie these regions in relation to the milk that mothers had made for them when they were babies. And what we further found, consistent with our predictions, was that early milk yield was very predictive for daughters, and peak milk yield and peak density was associated for sons. So this fit in perfectly, right? We thought that there was earlier sensitive windows for sons, later sensitive windows for, saying this all backwards. We thought there were earlier sensitive windows for daughters and later sensitive windows for sons. And oh my gosh, these are the first data. They're showing systematically robust associations between the milk that mothers made for individuals when they were babies and how their brains are functioning in adolescence. Now, can any of you spot what might be part of the problem here?
They're not breastfeeding all the way down.
No, they definitely aren't. They get weaned at like 6 months of age. But yeah, no, we think it has to do with organizing their brain. But when you see something showing up in almost every brain region, do we think it might be about just those brain regions? Or might there be something different going on globally in the brain? So what is actually going on is that the milk volume that's being produced is shaping whole brain glucose activity. And so we want to look, if we want to look at those regions specifically, we have to control for that background glucose uptake in the brain and control for their body size. Turns out you can't just look at the brain inside a vacuum from the rest of the body's energetics, even though a lot of neuroscientists do that quite often in the literature, but it's changing. All right, so what we did is we controlled for whole brain glucose utilization, and we controlled for the body mass of the individual, okay? Bigger animals are gonna have higher glucose use because they have more tissue. Okay? Now, maybe they're bigger because they got fed better in infancy, but can't really know that entirely. Talk about it. But our findings are a bit of a step back. So what we find is that it's not as consistent across both hemispheres. It is not as predictive for daughters as it is for sons, but we do still see that there are these different windows of sensitivity. Daughters are sensitive to early mother's milk and sons are sensitive to peak milk. So our findings are consistent. They aren't quite as dramatic as when I was first dancing in my lab. So, but in many ways, these are more exciting because they're real and accurate and we can believe in them. So data you can believe in. All right, so what we're actually finding is that rather than milk organizing the temperament in infancy and then that shaping behavior later on, what we're really seeing is likely that milk is simultaneously organizing growth trajectories, metabolism, temperament, and neurodevelopment directly. And we're seeing sex-differentiated windows and magnitudes of sensitivity, but not as important do we see temperament as predicting what the brain's doing later on. Okay. Now, we just looked at the milk energy, the fats, proteins, sugars, and volume of milk that's being produced. But we're going to go back now and look at things like cortisol. I'd really like to look at some of the lipids because these are the structures that build the brain, and other kinds of neurotrophic factors. Okay? And we're in the process of tying this in with how the animals are actually behaving in their home group with their friends. So takeaways, these correlational findings show consistencies with the literature spanning animal and human studies. Mother's milk contributes to neurobiological organization that may influence adolescent and adult function in primates. Sons show evidence of greater magnitude of sensitivity, and daughters show evidence of earlier windows of sensitivity. And I feel quite comfortable saying these things, in part because we've actually done a really rigorous control statistical analysis, but also because elements of all of this are already documented in the human and primate and rodent literature. So all the pieces haven't been brought together the way our study has, but they rest on a foundation of other established knowledge, which gives us greater confidence in our findings. Did I end with this picture last time? We didn't get to the end. We didn't get to the end. So this is a painting. This is Kandinsky. He was one of the early artists in the abstract movement of the 20th century. And he called this painting his the most complex painting that he'd ever made. And he spent many months painting it. And I look at this painting and I think this is barely the complexity that we see a mother synthesizing in her breast milk every day of lactation. And I find that an incredible triumph of adaptation. So thank you for your attention. And I have time for questions. I got to the end of my super shortened talk for once. So thank you very much. I know I didn't explain it that well. Please. Yes, thank you, Roseanne.
Daughter having that earlier sensitivity.
Yes, I might even actually have, almost like I planted you. Okay, so people are always interested. We find one of the most robust findings we have in recent areas of this kind of evolutionary focus book is that the recipe is most dependent on it's over and over. And it's not, I don't think that it's better for sons or better for daughters. What I think it is I think it really tailors the developmental priorities of sons and daughters. I think those two things. And one of the biggest things that's different between them is the timeline to adulthood. So we codify in our culture in ways that legally, what's the difference legally? So it's equally applied to everyone, but it's not. 18-year-old is a daughter. But if we look at like the trajectory of like reproductive debut, right? So having your first baby, it tends to be at younger ages for females than it is for males, in humans and other mammals, most of them. So this is what this was here. A rhesus macaque female has her first baby around 3.5, 4 years of age. A male's not going to enter reproductive maturity and sire an offspring until he's like 6 or 7 years earlier. And this means that fundamentally, mothers have a greater capacity to influence the adult condition of their daughters than their sons, because their sons have so much more time to either compensate for deficits in that parental endowment or squander what their mother gave. Whereas daughters, by that shortened time period, there's just greater effect size. And so I think that's a really big part of it. Another thing that might be going on is in rhesus macaques, daughters stay in their social group their whole lives. So they stay with their moms, their sisters, their aunties, their cousins. They form these like long-lasting like grooming groups. They interact routinely. Whereas sons at reproductive maturity are going to immigrate out to a new group. And so if like a higher volume of milk means that there's more frequent nursing, and that means that there's a different kind of bond established between moms and their daughters, part of that the tie that binds the female family network might start missing. And sons have this different behavioral dynamic with their moms. I'm not really sure that's a good explanation for it. You're not really seeing it in the data in terms of like a different behavioral breastfeeding structure. And the other thing is that we've done this work in dairy cows. This is so cool. So in dairy cows in the US, calves get pulled at birth. They get raised in a calf barn on milker freezer, and the moms are mechanically milked, right? And that like robot milker doesn't know that she had a son or a daughter. And what we find is systematically higher milk volumes when you gestate in the daughter. So it suggests that there's likely something physiological going on from a daughter fetal cell to the son fetus and how the mammogram tissue develops. But we don't know anything about it. Yeah, I agree. So there's likely a physiological component and a behavioral component. But I think the behavior component is going to be pretty nuanced, and I don't think we're quite at sample sizes in the monkeys that we can get there yet. Any other questions? Yes, thank you.
So you said you were interested in mobility or effect with its effective brain development.
Yeah.
What would you expect to see?
Well, I'm really inspired to the neurosciences. I'm really interested in the sphingomyelin. So sphingomyelin is part of the fat component within the milk, and the sphingomyelin form the lipid sheets around neuronal axons. Right? So they basically, the more insulation you have there, the faster electrical impulses run, and the more quickly your brain functions. And we know that there's really big differences in sphingomyelin between like formula and breast milk. We should talk about that, mostly because I'm like a neuroscience student, but I'm really curious to know, like we did have like kind of gross fat composition, but not with some parts of that fatty acid profile. So I'm really interested to look at. I'm not as interested in things like aging or motility or being addicted, in part because I don't think that some monkey models are right models to look for that. Humans get a really different omega-3, omega-6 profile because we eat a lot of nuts and animals and things like that. Whereas monkeys are just consuming a lot more fruits than we eat. And except in captivity, they eat Purina monkey chow. You guys know that? Purina makes a monkey chow. And so their diet is much more like a human diet because the protein content is largely going to be animal-based protein.
Purina doesn't have to tell us where it's sourcing. So this is really interesting. The National Institutes of Health used to, like, Purina will put animal protein into like the monkey chow and dog chows and things like that, and they don't have to tell you the animal source of it. So they're just buying whatever animal protein is cheapest on the market, and when they're making their batches of things. And so, like, sometimes that's going to be beef, sometimes it's going to be swine, sometimes it's going to be fish, and they only have to reach certain, like, minimal thresholds for some fatty acids. There's no maximal, right? There's not like a dangerous level. They have to stay below. And so some people have speculated that, like, years when the monkey chow was really high in like fish, they were like, I think the monkeys are performing better on cognitive tasks. No data. No data. Everybody's like, yeah, like, you collected samples of all the food the animals were fed every year, right? And like, no. Unexplained variance. Yeah. Good question. Anyone else? Yes.
I'd like this research is pregnancy, but are there any interventions?
Thanks.
Are there any interventions that respond to that's so that's fun?
That is a great question. It's a really good question. And because of time, I spent a lot of time talking about complexity of policy and action. Right? So, like, one of the things that I think this is really good for shaping policy like parental leave. Right? Like, we should have a parental leave in the United States, which we mandated by law. And also, I think that talking about like the hormonal component within milk, like formula is never going to have it's never going to be the food processing technology, and the way that is regulated as food is not going to allow things like hormonal, and that actually hurts, right? Well, I get parents that are like, oh, I'm really stressed, but I'm not breastfeeding my baby. They're like, no. That's the cortisol is not. Love is stress for you. Stress. And like that cortisol really is important for shaping all sorts of babies to love. I'm much more worried about when babies get none of that. We know that human babies have glucocorticoid receptors in their gut, and that they're highest intensity and they decline after their age. And the breastfed babies' glucocorticoid receptor density in their intestinal tract is 5.5 times higher than the those receptors are there to get a signal from what they're doing. And when what they're doing does have that signal to downregulate that physiology, you don't have any idea. So there's legal reasons why they won't let out of hormone, but like right now the formula industry is $44 billion. We expect the next by Kandinsky to be $75 billion. They can do their own R&D. And I'm more interested in trying to understand these kinds of complexities and messaging about them in a way that becomes useful information for shaping points of decisions insofar as they have this capacity and moving on. Not everybody has equal access to that today sport. In the US, you know what the average amount of time is for when they return to work after baby birth? How long is it? 10 days. 10 days. There are many women that return to work within 3 days of giving vaginal birth and 4 days of C-section. This is so horrible and immoral and freaking an assault on human well-being for everyone. Other countries look at us and they're like, why are you guys like literally so so I think that like we're not at a point where we have enough research to be able to actually influence intervention, but it's something that we start talking about. So one of the things that we got in terms of like clinical applications that you could start doing clinical trials on right now about any concern of moving consequences, and actually start coding when milk comes to a lot of babies in Indiana. They're feeding of mother's breast milk that they're not feeding the breast milk. Mom's constantly giving it to their ministry. Why don't you start feeding morning milk and morning milk? And what if that helps basically improve and sleep for well-being or things like that? You do things like that. Because at home, moms are pumping and freezing and stuff. That's not the kind of thing on their day. You wouldn't know how anyway they not.
I guess even during pregnancy, before the baby's born, stuff that you have on the mom that during pregnancy to change some of the factors that you're that great.
Oh, that's a really interesting question. That's a very interesting question. And I think we're going to see a lot more information come out about that in terms of the first 1,000 days, right? Because like anything you do pre-lactation is necessarily going to have some sort of impact on the food. And so finding ways to support a healthy gestation and lactation as like a whole unit is kind of where the whole is going right now. But it's getting a lot of questions. Good questions. All right, I think we're over time now.
Yeah, I think we're over time.